This video covers the process of meiosis. Meiosis is the process of making gametes. Gametes is our fancy science word that means sex cells. So sperm or egg. Either of those, the more general term is gamete. Meiosis has two steps, meiosis 1 and meiosis 2. And so we'll cover one and then the other. You'll notice that it's the same sort of general idea as mitosis, but with some differences. The end result of meiosis is 4 genetically different cells with half the number of chromosomes. So with mitosis, making 2 genetically identical cells, meiosis, 4 genetically different cells, but that have half the number of chromosomes. And what we'll move on to next is looking at diploid versus haploid, which is the terms we use to describe how many chromosomes a cell has, or how many copies. So a diploid cell has 2 copies of each chromosome. The prefix di means 2, so diploid has 2 copies of each chromosomes. Diploid cells are a product of mitosis, and we label them as 2N, where N is the number of chromosomes, so that's telling you there are 2 of each one. And diploid cells are our somatic cells or body cells. So any cell in your body, for example, is a diploid cell, except for the ones that make sex cells. A haploid cell has 1 copy of each chromosome, and it is a product of meiosis. And it's labeled as N. Haploid cells are our gametes. And so that brings up the question, why do haploid cells need to be made by animals or plants, etc.? They need to be made because as part of sexual reproduction, we have fertilization. And during fertilization, 2 haploid cells fuse together to form 1 diploid cell, and that diploid cell is called a zygote. So if 2 diploid cells fused, that cell would have 4 copies of each chromosome, and that's just not going to work. So as part of sexual reproduction, haploid cells are made so that when they're combined, it makes a new diploid cell, which gives rise to a brand new organism. So let's look at some examples. Let's say an organism's diploid number is 52. Then I know that its haploid number would be 26. Just to go from diploid to haploid, we're going to divide by 2. For another example, suppose that an organism's haploid number was 27. And we're going to double that to get the diploid number, and we get 54. In humans, the diploid number is 46 and the haploid number is 23. So all sperm and all eggs should have 23 chromosomes, and when they combine, when sperm and egg combine during fertilization, that new zygote cell has 46 chromosomes. Sometimes a mutation happens and maybe an individual in the human case would not have 46 chromosomes, and we'll look at those next week. So this is focusing on what should happen, okay, to get that 46 chromosomes for humans, or whatever the diploid number is for any other organism. Next, we are going to look at meiosis 1. Just as a note, interphase and DNA replication happen before meiosis 1, and it's those same details as when we talked about interphase before mitosis. The first step of meiosis 1 is called prophase 1. We have to put a number after it so that we know that it's happening here and not during meiosis 2 or during mitosis. So most of the same regular prophase things that we already know about are going to happen here with a little bit of a difference. So we know that our spindle fibers are produced by the centrioles. Something new is that homologous chromosomes pair up. A homologous chromosome is the corresponding chromosome from each parent. So for organisms, mom provides half of the genetic material and dad provides half, so that we have 2 copies of each thing. And so the homologous chromosomes are the ones that match up. Then a special thing that is unique to prophase 1 of meiosis 1 happens. That's called crossing over. During crossing over, homologous chromosomes exchange genetic info. So let's draw what that might look like. So here is a replicated homologous chromosome. This pink one is, we'll say, maternal, and the green one, oops, let's fix that. The green one is paternal. And during crossing over, what happens is these homologous chromosomes are going to get kind of like really close together and they get all tangled up. And our end result of that is that the chromosomes will be like a combination of the two. So this is one of the reasons why the haploid cells that are made are not genetically the same. It's because crossing over happens where the homologous chromosomes are going to exchange bits and pieces. Okay, so now each chromosome is a combination of maternal and paternal DNA. And then spindle fibers are going to attach to the homologous pairs, and there is a picture. I know it's a little blurry to you, but that's because I've zoomed in. On your copy of the notes, it's going to look just fine, and we'll diagram this together later anyway. Later meeting in class. After prophase 1 is metaphase 1. And during metaphase 1, homologous chromosomes line up in the middle of the cell. In the middle. So in mitosis, they line up single file, but in meiosis, they line up with their buddy. Going to line up with their homologous pair. And then after metaphase 1, we have, you guessed it, anaphase 1. During anaphase 1, the homologous chromosomes are pulled apart, which you can see here. And I hope you'll also notice that on these pictures, after crossing over, the chromosomes have bits of red and blue, okay? And that's because of crossing over. And then telophase 1 happens where it's normal telophase. The nuclear membranes will reform around the genetic material. And then we're ready for cytokinesis, where the cytoplasm divides, which you can see there. And then these two cells that were made in meiosis 1 go straight into meiosis 2. In meiosis 2, we're going to start off with prophase 2, which has all of the normal prophase things that we've ever talked about, except here there is no crossing over. So crossing over only happens in prophase 1, not prophase 2. And then during metaphase 2, the chromosomes once again line up in the middle, single file, which you can see here. And then in anaphase 2, our sister chromatids are pulled apart. So our replicated chromosome looks like this. Each little arm or each little piece is a sister chromatid. So those are going to be pulled apart. And then we have telophase 2, where our nuclear membranes reform, and then cytokinesis, where the cytoplasm divides. And we'll see a picture of that all the way over here. That is our after telophase and cytokinesis. So to sum up the whole thing, interphase happens before meiosis as well. We also know it happens before mitosis. Meiosis starts with one diploid cell, and it ends with 4 genetically different haploid cells. We also know that a new diploid cell forms during fertilization. And that this whole process is part of and necessary for sexual reproduction. And that is our brief summary of meiosis. We will look at this in more detail in class.